Hi everyone. Vignesh. Hi Vijay.
Hi, Manshu. How are you?
Nice. What's the time now for you? It's like 10.
9 o'clock in UK. Yeah.
Oh, 9, yeah, 1:30. It's 5.5 hours behind, right?
Yeah. Mm.
Okay, cool. I see quite a few students here. That's good to know. Thanks.
That's already
Yeah.
early 7.
So I would like to start by thanking Vijay again. To be kind enough to actually host and conduct this half a day workshop. And I think Vijay, you were mentioning one of your postdoc, right?
Yeah, he has joined us. Anuj is here.
Oh, hi Anuj. Hi Anuj. Welcome. Thanks for quickly taking over what Vijay was supposed to do. But yeah, but thanks to Vijay for sort of suggesting this and organizing this workshop on single cell RNA-seq and other single cell technologies. And I've been told, Anuj, that you are an expert in doing these, and you're carrying all the good work that Vijay claims to be doing. So, so I would say that I think we hope that many of the students who are here would learn a great deal about the various RNA-seq or single cell RNA-seq technologies that have come up in recent years and how they have been transforming sort of the way we understand biology at this sort of at the heterogeneity in various systems that we see around, even though we try to now even though we don't have enough information at the organism level, but I think these heterogeneities are leading to several new ways of looking at diseases. And I think especially what the focus of Vijay's lab is cancer research, where you are actually looking at how cancer cells, different cells within a cancerous tissue, respond to different things, and that can therefore lead to ways of manipulating and hopefully treating cancer by targeting very specific set of cells within a cancer which become recalcitrant or which become the reason why recurrence or remission might go away. So I think I would not take too much time and then welcome Anuj Dwivedi again to the platform and hand it over to him to take forward the workshop today, and Vijay for accepting this, and they finally are requesting Anuj to, I can understand various other commitments Vijay would have. So it's always pleasure. It's always good actually when postdocs do it, because then they get trained. And then that's one thing that I think Vijay would also really appreciate. So yeah, so VPIs can move out and let the postdocs and graduate students interact, right? Without our Sauron like eyes, right?
Yeah, absolutely. Yeah. Thanks, Himanshu. So, hi guys. I see a lot of you around here. I see already 90 people in the call.
Yeah.
That's excellent. That's excellent in number. So how we have organized. So I'm going to give a short talk to begin with. To give you introduction of why single cell is important and how you can use single cell data to understand various aspects of biology. And then after this talk, I will pass it over to Anuj, who will conduct into the workflow of how to retrieve single cell data, how to process, what are the kind of things you can do, how to design an experiment, how to analyze and interpret. And he will also do a hands-on workshop where you will analyze your own data. Because this, and remember all students that this is not a classical teaching class. This is an interactive session, and it should be like this throughout the day. So please feel free to pause, type your questions, stop the lecturer whenever we are tutoring in case you have any questions or confusion. So let it be an interactive class. Otherwise, the purpose is not achieved. So I will go on to do a small presentation to build in excitement of why this is important. And with this fire, I will pass the bomb to Anuj. Okay. Yeah. Is my slide good?
Yeah, yeah.
Okay, so I'll give a brief outline of why we think it's now possible to decipher biology in high resolution through single cell genomics. To put things into context, imagine the very fundamental process of development. Our life begins here when one oocyte fertilizes with a sperm, makes a zygote that undergoes lots of cell divisions and forms the structure called blastocyst that goes on to form embryo that eventually develops into an infant. What is obvious is that from this single cell, we generate several different tissue types in our body. And each of those tissues have identical genetic material, but have different gene expression program. So in other words, our development and our adulthood is nothing but a consequence of differential gene expression program. Because if our tissues would not express these genes differently, all tissues will have same program, and that would not work. Our liver has different set of genes expressed, kidney has different set of gene expressed, and brain has different set of gene expressed, so on and so forth, despite having the identical genetic material. So it's the differential gene expression program that sets the cell identity. That's an important point, okay? Now, as we grow into an adulthood, we are also exposed to a number of external signals, be it different types of diets, the circadian rhythm, the way seasons are responding, when we are exposed to viruses, toxic chemicals, drugs, and so on and so forth. And we have also learned that over the last years, that all these external stimuli, they also eventually manifest by targeting gene expression program. And last but not least, many diseases are now shown to be a consequence of altered gene expression program. For example, in a normal situation, this is a tumor suppressor gene that's active. It protects our cells against carcinogenesis. While in a cancer cell, the gene gets inactivated, in this case, for example, by DNA methylation, rendering a tumor suppressor gene inactive and thereby causing onset of oncogenic events. So to appreciate, you could imagine that diseases are a consequence of altered gene expression program. So irrespective of the systems and the projects you work on, you are eventually dealing with differential gene expression programs. Thankfully, we are currently living in an era where a number of genomics techniques have advanced, and they allow us to look at different facets of this process. For example, if you want to map chromatin or understand epigenetic modifications, you have a number of methods as listed here. If you want to look at DNA methylation, you also have distinct methods to interrogate. If you want to understand the genetic identity or genetic sequence topology, you have whole genome sequencing, exome sequencing, chromatin confirmation for looping, like Hi-C-like techniques. You can also probe gene expression through classical bulk RNA-seq approach, and today's focus single-cell transcriptome, and now even we can do spatial transcriptomics, where we do not need to dissociate the tissue, but map gene expression programs in the intact 3D context. And now you could combine these genomics methods with various perturbation methods. For example, you could combine CRISPR perturbation techniques together with single-cell genomics. And eventually, the most powerful way as of today is actually multi-omics, where you combine one or more of these methodologies to understand various aspects of cell fate. Classically, we used to do a lot of bulk RNA analysis. So what we did in bulk RNA, basically, if let's say we have a heterogeneous tissue, you could imagine there is no single tissue that only had one kind of cell type. So what you would do if there are different types of cells within a tissue, for a bulk, you will grind all this together. And when you do so, you generate an average gene expression of all cells. So eventually, if you observe something, you do not know, is it coming because of a dominant cell type, or is it coming because of an altered one? So we always masked a lot of information, in particular, those coming from minor subpopulations. Now, so single cell, when it came, it was also named the method of the year by Science magazine because the power is unprecedented. You could imagine taking each cell at a time here and performing transcriptome analysis for all of these cells individually. Then eventually what you do, you ask the program to cluster each of these cells based on similarities in their gene expression program. So you would eventually see a pattern like this. These cells, even though they were individually profiled for transcriptome, they clustered together here. That means that they have identical gene expression program, and therefore likely they are the same cell type. This way, in this single experiment, you have transcriptome of each cell type, each cell, and then you have identified at least 4 different cell types within this tissue. So studying heterogeneity has been the most powerful application of single cell methods, where if you have different cell types or there is a different in the gene expression program within this tissue, one is able to identify this heterogeneity. In a developmental context or in a progression context, for example, moving from low to high metastasis, it's also possible to perform what is called lineage tracing study, where you can organize the transcriptome of these cells in a pseudo time course, where you would say, okay, maybe the cell began here and had this trajectory and versus this trajectory. Some systems biologists are also using the single cell data to model stochastic gene expression program, because some genes are expressed in on-off fashion and not in a linear mode. I'll highlight one example of this process. For example, we work on what is called triple negative breast cancer. Triple negative breast cancer is 15 to 20% of all breast cancers. And approximately half of these females will develop chemoresistance, which is pathetic because that would mean we have no options for these 50% females. We have to just wait for deathbed. So they have very poor survival outcome, 12 to 13 months for metastatic TNBC. And the costs are very high. So we thought, okay, there is a way to actually predict this chemoresistance in these females upfront before they undergo chemotherapy. And the hypothesis was that the patients that are resistant to chemotherapy have some of these bad cells present within their tumors. And if that's the case, when you apply chemotherapy, the normal cells will die off, leaving these resistant cells intact. And that will again repopulate despite exposure to chemotherapy and metastasize. So pre-existent of a chemoresistant population underlies chemoresistance. And eventually, our postdoc Inayat performed this analysis on single cell data sets of pre and post chemotherapy in resistant and sensitive patients. And he identified that in the chemoresistant patients, in the single cell RNA-seq data, there is a distinct population that is only occurring in chemoresistant females. And this population was enriched for so-called EMT or epithelial to mesenchymal transition program, which is known to associate with high rates of metastasis, chemoresistant stem cell properties. We went on further to derive signature genes from this population and build a model that is predictive of chemotherapy and validated this on 446 patients. The patent has been filed and accepted in UK. So in that setting, we have now a 20 gene signature that by RT-PCR would be able to predict in these TNBC females upfront before they undergo chemotherapy, whether you are likely to be sensitive, that means responsive to chemotherapy, or resistance. And now we are also using this gene signature to actually see whether we can drug repurpose some of the drugs to convert the resistant females into sensitive. We also use single cell to understand other aspects of cell biology of metastasis. For example, Ryan Lusby, a PhD student in the lab, he performed a pan-cancer integration of single cell RNA-seq data set of all major cancer types: breast, stomach, colon, kidney, liver, lung. And by integrating this, he was asking whether there are any common hallmarks of metastasis that are shared across all metastatic cancers. He has successfully done this, and now he has been able to find that Wind signaling is a common core marker of metastasis across a variety of cancer types. Another student in the lab, Emily, she has been looking into understanding using single-cell RNA-seq data to understand when the cancer cells are migrating, are they switching into different types of cell migration? And she is able to find that indeed there are at least switching between amoeboid and mesenchymal phases as cells transit from a benign to a metastatic state. Another student, Aoife, has been looking at single-cell RNA-seq data sets of autism spectrum disorder patients. She has compared single-cell RNA sequencing data sets of ASD and healthy individuals, healthy versus ASD plus ADHD that have both, or ASD plus epilepsy or ASD plus depression. Using this analysis, she finds that one of the most commonly disrupted pathways is synaptic signaling, neuronal differentiation, and neuronal development. So you could imagine that using single cell data, we identify that across these comorbidity disorders, the common pathways that are affected are related to neuronal development and synaptic signaling, which makes a lot of sense. These were some examples. So I have outlined some examples of what all you could do in a single cell data. For example, you have a heterogeneous tissue, you derive single cell data, and then you could actually identify a signature gene, as I've shown you earlier, that associates or defines a cell population associated with a pathogenic state. Now, using that signature gene, you can go back to some existing databases. For example, TCGA, you might all know. You can go back and look how that signature behaves in TCGA. There are also now other databases such as DepMap, where you are able to look at what would happen if these genes are perturbed by CRISPR and whether certain cancer cells actually require these genes for their survival. Then you could look at their prognostic value and consider them for stratification or model building. Once you perform clinical validation, you could eventually consider a commercial kit for diagnostics and launch this in the market. Second possibility is use this data to understand the mechanisms or pathways of diseases. So let's say you have single cell data. You could do what is called cell type regulatory network analysis. For each of those cell types that are detected in single cell data, one is able to draw interference on gene-gene interactions and which genes co-vary in expression and dominate each of these cell types. One could then also do looking at biological processes and pathways that are active within each of those cell types, and eventually generate models of certain transcription factors that govern these cell type associated gene expression programs, and then go back to these databases for perturbation analysis and further validations. Another possibility is into drug discovery and drug repurposing. For example, you obtain heterogeneous tissue and looking at, for example, responsive and non-responsive patient, and you find that here is the gene expression program that's specifically associated to a non-responsive patient. You could go on to a signature gene validation and then use certain databases to understand the relationship between gene expression versus drug sensitivity. Then one could build a model for therapy response and check whether we have possibilities to repurpose alternative medicine against this drug, and therefore move further with clinical trials and validation. So you could see that single cell actually data allows you to, single cell transcriptome allows you to have enormous possibilities to understand the mechanisms, to understand diagnostic biomarkers, to identify new targets for therapy, and obviously to give a bigger picture of certain cell states of perturbed to cause diseases and confer different aspects of disease pathogenesis. So with that, I want to thank you guys. I'm happy to take questions before I pass on to Anuj for holding the workshop. Feel free to ask.
Good afternoon, doctor.
Yeah, Abhay, go ahead, please.
Yeah. Doctor, you were talking about the resistance to the chemotherapy for triple negative breast cancer. But I wanted to ask you, is it the only treatment modality? Because primary treatment includes mastectomy before you go for chemotherapy. Of course, if it's metastatized to different organs, then chemotherapy is must. So in that case, but since it's a primary cancer in breast, then removing the breast itself is a primary treatment, and then we go for chemotherapy.
So I'm referring to mostly to cases where metastasis has happened, because this is a major problem, right? So once metastasis happens, you have no other chance than doing chemotherapy. If you could detect at an early stage, of course, you would do the dissection as you mentioned.
Thank you.
Yeah.
Good afternoon, sir.
Hello, Galivati.
Yeah, chemotherapy will be the solution, no doubt. But what are the chances of getting cured? As we read many times, chemotherapy even affects the healthy cells surrounding it, right?
Sure.
So
Right.
how do you
Yeah.
suggest it is mandatory for the chemotherapy something?
Yeah. So you're absolutely right. Chemotherapy is not discriminating tumor versus healthy cells. And therefore, there are now alternative approaches in development where you would be able to target specifically the tumor cells. One of them is triggered what is called antibody-based therapy. So you would target a surface antigen on a tumor cell, and that antibody is coupled to a toxic chemical.
Okay.
So these things are in development. They will come in future because this is recognized that chemotherapy is basically killing everything.
Yeah. Thank you, sir. Thank you for giving insight into the new development.
I think Vijay, we should. It's already close to 2. Maybe we can. Anuj will be able to answer some of the other questions that would crop up.
Yeah.
Right, Anuj. So maybe we should begin so that we can. We'll have some. Anuj, do you have a plan that you can share in the beginning so that people have an idea how long are the sessions, what are the break times, and something like that? Or
okay. So thank you, Himanshu, thank you, Vijay, for giving me a chance to conducting such a workshop in larger scale. I could imagine last time I conducted such workshop on some other central universities. So my whole slides and program are comprised in 2 parts basically. First one is that where I will be introducing these methods, what we are using, and giving hands-on and telling what are the different modalities we are going to analyze. So giving data, already have shared the data. So if you people have got the email, so I hope you have downloaded the data and installed the softwares. Okay. So first thing is that I will show you that how to do that and what are the different commands to use that. So once you do that, we will take around 45 minutes break after that, and then I will come back and analyze the data, each and every aspect of the data, like from QC, then scaling, then normalization, then clustering, then annotation, then differential expression. So first you try to analyze the data by your own, by whatever the commands I have provided to you, and I will provide meanwhile the commands for the each steps. You first try by yourself, okay, and then we will discuss all the output of each steps with the everyone.
Okay, great. Okay, I think Anuj is very well trained, Vijay, to handle this. Maybe this is slightly larger. We have what, 126 participants. That's we exclude ourselves, so 124. So, but good luck, and so maybe Vijay and I can log off and then hand it completely over to you, Anuj, to continue.
Yeah. Have fun, guys.
Yeah, have fun. Yeah. Okay, bye, Vijay.
Sure.
We'll be in touch. Yeah.
Yeah.
Bye.
Yes. So hi everyone. So let me introduce. Already we have introduced about the single cell RNA-seq technologies and the applications and the other different modalities we have analyzed in our data. What are the applications we have used for the single cells? So I will start with the first basic introduction. Then I will go to the technicalities of the single cell RNA-seq analysis, and then the tutorial for today. Okay. So I hope this is visible to everyone.
Yes,
Yeah.
Anuj sir.
Yes, thank you. So, guys, welcome to everyone for this workshop. Today we are going to discussing, and I will tell you about the mainly methods and the application analysis pipe we are using, and mostly used by most of the people for those who are analyzing single cell data. Then you can realize the importance of single cell when it comes first time into public space, that it be named for the method of year in science was when the single cell come. Then consequently next year it was named method of nature methods of the year, because when they introduced the single cell multiomics, like single cell attacks with the single cell RNA-seq. Then next year again it was nature method of the year, because they introduced the single cell spatial transcriptomics. So you can realize the how scientific world was benefited, and then consequently it was again back to back nature in the big magazines and the top journals. Okay, so let me just give you a brief overview why single cell is important in the layman language. That if you have the complex, like suppose you have a bunch of a bowl of different fruits, okay, and then you suppose prepare a shake, okay, and somehow some people like it, some people don't like it, okay. So in the bulk genomics you can just know that this is shake, okay, but you don't know what is comprised of. Like some people like strawberry, some people don't, like some people like grapes, some people like peach. So single cell gives you leverage to understand what are the different fruits and components contributing to this larger picture of the shake. Okay, in the bulk genomics you can just see the shake, but in the single cell we can actually really get to know what are the different components contributing to the shake. So similar scenario in this our body also. Okay, there some cellular heterogeneity is present. Okay, for example, here you sequence one tissue and you took the one tissue and sequence it, and then in the bulk you see the three genes are expressing a different level. Okay, this gene is expressing at lower level, this gene the more higher, and this is the moderate level. But actually you don't know. I think, could you please mute other people? Okay, so if you see the genes having the moderate expression have the different set of cells where these genes are expressed, and the genes with the higher expression have the different cell of there where it is highly expressed, and the genes with the lower expression is highly expressed in some cell types, which is being ignored in case of the bulk samples. Lisa has already mentioned this, how this cellular heterogeneity is being measured and identified in the single cell data, which is missed out in case of the bulk. Then let me tell you the methods. So this is the typical workflow is comprised of our wet lab and dry lab analysis experiments. So it starts with the solid tissue. You dissociate the cells using different methods available in the wet lab, and then you isolate the single cells. Then according to your questions, I have also highlighted just single cell DNA and RNA and DNA methylation sequencing, but there's a lot of other sequencing you can do. It is up to you what you want to sequence. Okay, so there's a like once you prepare a material from the starting point, like your sequencing, then it is just a simple bulk sequencing. You have to just feed these input materials and like sequence it. Once you have this sequencing is done, okay, so it is like in the case of bulk we process it separately. In case of single cell we have different methods and a pipeline to proceed further, and ultimately we want to understand the transcriptomics and their proteomics changes at a single cell level. Okay, then there's a different methods to capture the cells and library preparations, but one of the most used methods, which is widely used and most advanced one, is the dropsic methods, where different drops are bypassed and then emulsed with the oil, and this oil captures the cells and then microparticle attached to them, then do the RNA hybridization, And then we perform the sequencing by adding the different barcode to different cells. Okay. And then these barcodes are added, and then again PCRs, and then sequencing and the parallel PCR happens. Okay. So this is the dropseq method, same method used for the 10x. 10x is the most widely used single cell sequencing platform, where it starts with the isolating the cell. First, they freeze the cell, then sift the samples if you don't have sequencing facility at your own building. Then they check the viability of the cells. Is very important because you don't want to sequence the dead cells. Okay, but in some cases, like when you are dealing with the nucleus samples, then you have some other way to check the quality. And then you feed the samples to 10x machines, and then they again start with this cell drop method, which I explained you earlier, that they start with the barcoding and then dropping everything. Then you get actually this sequencing and library preparation, the sequencing, normal sequencing, the reads are generated and everything. So already we have mentioned some applications to you people, but I would like to give some more examples in the area wise. So it was the first time in the neuroscience when people were able to detect different neuronal population. Even in neuron, there was two subtypes, GABAergic neurons and glutamatergic neurons. And apart from neurons, there was the glial population where they were able to identify the microglia, oligodendrocyte, OPC, astrocyte for the first time. Okay, so single cell enabled to identify these neuronal population at cellular level, and it was the first time identified in the mouse primary. Rasmi, could you please mute? Is there anyone who is controlling the box up so they can mute the participants and can be unmuted only on the request? Okay. Rasmi, could you please mute yourself? Okay, sorry guys. Then another example in cancer where there's a first time in people were trying to understand the cellular heterogeneity in the pancreatic ductal adenocarcinoma cells. Then it first in the anyone knows him? Knows her? Okay. So another example from the cancer where first time in the case of pancreatic ductal adenocarcinoma cells, first we were using the single cell, people could understand the cellular heterogeneity present at the PDAC, and then also understanding from pre-metastasis to metastasis cells, evolution of PDAC, then micro environment for the different cell types like fibrous plus, for example, then response to gene targeted therapies, then circulating tumor cells and other cells, and cellular heterogeneity overall. Okay. Then another example from the immuno-oncology. So here, this is again extension of the what different kind of single cell sequencing you can run using for the single cell data, but there's a different modalities which can be analyzed in single cell data. Like one is the intramodalities, and then the second is the intermodalities. Okay, so if you are analyzing the single cell from the different samples, okay, so then it will tell us the cellular heterogeneity and the subpopulation and everything, but there will be limitation because you are not able to identify some other cell types present in the different samples. And also when you are integrating the samples, you might not be able to get to the what is the transcription like epigenetic effect when you are just integrating one modalities. Okay, the second thing is you are integrating two different modalities like single cell RNA and single cell attack, which can actually tell you one more thing, that apart from the expression, you can also get to know what are the transcription factors binding to different regions and how these transcription factors are changing the expression at the cellular space, especially different cell types. Okay, so that can be like transcription factors. Biological network can be understood in a detailed manner when you are actually integrating the single cell with the tag. Okay, then another is that you are actually generating the same single cell attack from the same samples. Okay, then this will this tells you, because this is generated for the same source, this is single cell multi-omics, so you can infer the same TF regulation networks but in a both effective way, because you can also understood the cell to cell communication, and also you can do the linear tracing in a detailed way. Then personalized cancer therapies, as Vijay mentioned, we can use the response of the different patients into say a single cell level and how they are responding to different therapies, and then using the different database, we can actually predict the personalized drug. Okay. Then in the clinical trials, it is always very expensive to use single cell in clinical trials because it took a lot of money, but it can be done once you have the selected population, some target population where you really want to know the impact of your drugs in a depth. Okay, so like you selected some population and you did some single cell analysis, then you understood the immunotherapy, then immunoresistance. So rather than conducting the experiments on whole population, you just select the set of the population and perform the single cell, and then you really get to know what is happening at molecular level, and then decide your outcome of the clinical trials. Although again, it is taking a lot of money, but gives you more detail. Then analysis is comprised of different portions like sample preparation, then cell capturing, then reverse transcription like PCR, then library preparation, then sequencing, then analysis. Okay, so this is a typical primary analysis which we do, and generally people have done using the single cell data, like identification of the different populations. Like first time you are sequencing some organs or some tissues, so you don't know what the cell types present in there. Okay, so you can identify really new cell types present in your tissue of interest. Then second thing, that once you are generating any organ specific atlas, suppose for the mouse atlas, suppose for the human atlas, so you can really get to know what are the different cell types present in the different organs. Like for example, what are the cell types present in the liver, and what are cell types present in the brain. Okay, then another thing is that if once you identify the different cells are present there, then you can also get to know how these cells are communicating to each other. So the basic principle behind this cell to cell communication, that you identified and map the genes which are responsible for the ligand and receptors. Okay, so we identify the genes which are what are the expression pattern of the genes responsible for the ligand and receptors, and then on the basis of the expression, we actually predict the cell to cell communication. Then for the developmental samples, and even some case of the disease sample also, we could infer the lineage trajectory, how this lineage how these different cell types are developed. Okay, first thing, in the normal cases, like developmental cases from prenatal to postnatal development of different cell types. First thing. The second thing, in case of disease, how this cell type trajectory and cell evolution changes under the disease condition. Then personalized medicine, again, I have told you the example in the previous slide only. Then spatial is something very exciting and very like important aspect, which is still people exploring. A lot of new technologies coming, Visium, then Xenium also came recently. So here people, rather than going into the detail of cell, you freeze the sample and then freeze the tissue, and then get to know what are the different transcription factors, what are the different genes are active, and cell population presented your slide. Okay. Then one thing is that integration of single cell multi-omics data. Okay, so there is two modalities, like one for the modality one is a single cell RNA, and then modality two is like single cell attack set. Okay, then first thing that you just simply integrate by them the expression of the gene or protein. The second thing that you can actually calibrate them at up to some level, rather than just taking their expression, you can harmonize them and then integrate them. Third thing is that you plot them in a different space, and then try to match their expression label from the different data sets, and then integrate them together. The third thing is that by integrating, comprising all the ABC mentioned above, and then comprise and integrate them, and then consists of 2 cells, and identifying the networks present in those cell types. Then we can also do, there's a very good advantage of, like, we have already done a lot of bulk sequencing in the previously, okay, before single cell was in the picture. So like we could use those sequencing samples also to go back and check what are the different cell types present in those samples. Suppose you have a lot of brain samples which have been sequenced, a lot of kidney samples which have been sequenced. So now you know what are the different cell types present in the brain tissue, what are the different cell types in the kidney tissue. So you can actually deconvolute the level of different cell types present in your bulk samples, like using the transcriptome profile of different cell types. Then you prepare this gene matrix having the different cell types versus the gene expression. Then you use the bulk data, and then permutation and combination. So it uses different machine learning algorithms to predict the different cell population in your bulk data. So you have to always decide these machine learning method based on the like statistical significance. Okay, so and that too up to more extent, it is also, you should have an idea that what it is the expected population of the different cell types present should be present in your bulk data. Okay, so on the basis of that, you can decide the best one. Then for the spatial reconstruction using single cell data, so spatial data is still on the developing phase, where people are actually understanding the fixed tissues expression and the expression in those cell tissue samples. So it starts with like you have the fixed samples there. So you actually modulate and predict the gene expression on the, like suppose you have this cut of the slice. Okay, so in the slice, you know, there's a different in X, Y, Z, which are expressed at the corner of cells. The genes which is also expressed in this portion of the like upper portion, and this in the right portion of the samples. Okay, then you use this spatial samples, then predict the localities of the genes, and then also on the basis of their expression pattern, you define a, like, this is the particular location of the gene. Okay, on the basis of expression. Similar things you also getting from the single cell data, where you have the gene versus cell matrix. Then you apply some machine learning methods, and then cluster them on the basis of the expression types, like these all 3 genes like X, Y, Z have different expression in the different cell type specificity. Okay, so once you have this cell type specificity from the single cell spatial data and the single cell RNA-seq data, then you integrate them in the bulk samples, and then map them how these different genes are expressed at different portion of the slides, and what are the expression level. Okay, so today we are mostly going to discuss regarding this single cell library analysis pipeline. So this is the pipeline which we are not going to use today, because this is the pipeline you are supposed to run when you get the data in the raw data in the single cell data from the 10x or any platform. Then it starts with the basically reads. They provide you the reads. Then you find the valid barcodes, which information already provided, because when you're mapping them, you provide different barcodes to different cell types, you know already. Then identifying, mapping the reads, then subsetting, then all these QC steps are done. Then ultimately we get the UMIs. Okay, so this UMI matrix is your main input for any further analysis. Okay, this need to be very this need to be understood, very important here. Okay, so this is the typical pipeline available in the 10x. Okay, so there is nothing need to be very going to detail of this pipeline, because this is already standardized and optimized. The most important thing is that analyzing the output of this files, the EMI matrix. Okay, so in single cell data, we have different file formats. Okay, so people store the different file formats according to their comfort, according to their analysis, according to their computational resources available. Okay, so like for example, people are storing SD5 format, then RDS. The RDS format is one where you have already analyzed your data, and then you want to restore the every aspect, like from the differential expression, from the scaling, from the normalization. You don't want to repeat it again. So you save it as an R data format. Okay. And then just every time, whenever you have saved that, you have to just call your R data format RDS file. Okay. Then S3 file, AD file, and Loom file. Loom is the most used format in the public data sets. So most of the public data set you will find in the Loom format, even the large data set are present in the Loom format, and in the MTX format, the matrix. So here, the matrix is the one you find mostly in case of the people, those who are submitting data in Geo. So here you can see how it looks like in the Loom format. Okay, so here in Loom format, you can see the different features, different of the cell types are present here, and the different classes. In case of SD5 format, this is again the genes, but in the different cell types. Okay. Then this is the S5 format. Okay, so in S5 format, one thing is that you can also get to know what are the different modalities. In addition to gene expression, like antibody capture, multiplexing is present. Okay, so here we start with the analysis. Okay, so I am just showing you this PPT right now, but I will also demonstrate the live example. Okay, so just to give you an idea, I'm just running, but I will run the live example. Okay, so first thing is that when you're reading the data, you just have to define the minimum cells and minimum features in a matrix you want to take. Okay, then here, like here, we have I defined the taking the genes at least have 3 expression in 3 cell types, 3 cells, and then also the cells which have at least 200 genes. Okay, once you supply your matrix, then the first thing is quality control. Then first quality control is the plotting the number of features, number of counts, and percentage mitochondria. So your N feature should be have the lower number of identity for the N feature counts. Then also all the cells should be might be clustered here, and then most of the percentage mitochondria genes should be also clustered here. So as a good cell, filter poor quality should be filtered out once. So to improve this quality, one step ahead, we can actually find the correlation between the features and the gene RNA counts. So the ideal case should be that there should be no cells should be present either in this sphere and this sphere. Okay, the most of cells would be this straight line and present here. This shows the good quality cells. Then next step is that we identify the different genes which we are going to use. So typically we select the 3000 genes. Okay, out of, like, for example, here out of 17,000, I have taken just variables for 3,000 genes, and then identifying the different, highlighting. You can also highlight what are the different top variable genes in your samples. Then you cluster them and scale them. So if you do PCA, in case of PC, you get different components. So you can select the component on the basis of the standard deviation they're covering. So it may vary most of the cases, but it may not vary. So the standard one is that you can see here, like taking 1:15 component might be covering all the components present in the data. Okay, sometimes might be just 10. So the next step, we do the clustering. Okay, so we try different component like DIMS, 1:15, 1:10, 1:20, and then different resolution. Okay, so the resolution and combination of different components define a clustering. Okay, so like here we have taken the resolution 5, where you can see the clusters are differently clustering different cells into different 9 clusters. And here at resolution 1, it is clustering different cell types, but not defined and expected as a biologically also. And here we are getting just very small number of cluster 4. So getting too many clusters and getting very less cluster, both are not good. Okay, getting the optimal number of clusters is the best one. So on the optimization episode, you can optimize the resolution and components here. Like for me, resolution 0.5 is looking fine to me. Okay, that's why I have taken this for further analysis. Then once you have these cell clusters, then you try to annotate them on the basis of the marker genes, which I'm going to tell you about in my this hands-on. And then you define them like NK, CD8, then memory CD4 cells, then pallets. So this is thank you, Vijay. I mentor. So guys, how would you like to start? Okay, so we can be interactive.
Hello, I just have one question
Yes.
regarding UMAP and PCA. What's the difference? Is that both are different or just same?
What PCA? What like which PCA are you comparing?
No, you showed the plot, PCA plot, and then UMAP plot.
Yes. So
So
PCA and UMAP are the same clustering path methods in nutshell, but there's a difference. So in PCA, the cells are not clustered as much as you can see in the UMAP, because the reduction methods used in the background is the difference. So in case of UMAP, you will see more clear clusters as compared to PCA. Okay, so I will also show the PCA plots where cells are not clustered very together. Okay, but in case of UMAP, you get the different clusters. So it is a UMAP improve the clustering, where in case of
Okay.
PC, you don't get such clusters.
Okay. And you mentioned that
Yes, Gauranga. Guys, please, could you please tell, I know we are, I think, around 100 plus participants. It is not possible to tell everyone. So do you have guys installed softwares which I asked? If anyone of can tell me.
We haven't. Can you just tell us again?
Okay, so guys, if you are online with your laptop, so just Google it and install R.
Okay.
Install what? Can you repeat, please?
R. Definitely simple R.
Anuj, Anuj,
Yes.
can I use the Google Colab for the R?
Yes, you can use. So you can use any platform wherever you can access R.
Okay, fine. No need to install. Thank you.
Yes. So see, you can use R anywhere, wherever you have installed. So let me share my screen so you can get to know much. Did you see the screen? No?
Yes, the screen is visible.
Okay, great. So guys, please let me know how many of you have installed R. You can just message me on the chat box.
Anuj, your version of R is 4.2.2. Is that right?
Yes.
Yes, thank you.
Good. I can see not many people have installed. Good. Very nice. So the guys who have installed the R, can you please install these three softwares packages in R, like DPLYR, Sirat, and then Patchwork, like this one I have highlighted. So I can do one thing.
We can use R directly from Anagana, right?
Yes, you can. See, I have just told you people, you can access R anywhere and wherever you want to do that. Okay, whichever is comfortable, you can use that. That's not a problem. Okay. My simple concern is that you should be able to run.
I wanted to ask, so you
Yes.
said that ultimately cell types are decided based on the marker genes, right?
Yes.
For let's say a particular cell type, or let's say cancer cell type, we don't know the marker genes. So is there any other way to classify them?
Okay, if you don't know the marker genes.
Well, it's not been reported in the literature, if that is the case.
Then how will you define the new cell type if you don't know?
That's what I wanted to ask.
Yeah, but yes, you are right. But my question is that if you don't have any marker genes available, then we can't do that. because this is all about the genes. And in a single cell, we are not even getting some, I think, in case of bulk, we get a lot of transcription factors. We get a lot of new de novo assembly and everything where we can map and annotate the de novo assembly. But from you can do one thing if you are working with such sample where you don't know anything or the samples like orphanage samples. So better to start with the bulk and then going to single cell. From the bulk, you get to know actually what are the different genes by annotating those genes using, you can do the blast or anything, and then going into single cell for those samples would be benefited. Okay, so
Anuj sir, I have a very rudimentary question in a
yes.
sense that if anyone wants to make a career in analyzing the transcriptomics, then what is the path to do it?
So career in what terms, like how
Qualifications are required for this?
Qualifications for analyzing the data most of the time, like in India nowadays, we have more defined this bioinformatics courses where people teach you all these analysis, but most of the learnings are by yourself, and you can refer these workshop which we are doing. Okay, so in most of the problem is that this is all mostly not defined so far. If we talk about India, okay, people in most of the university, you won't find people teaching all these advanced methods. Okay.
But there are university courses offering specialized courses in bioinformatics.
Yes, but the thing is that what are the level of their bioinformatics course? Okay,
So in general, they are teaching everything.
so like what they are teaching? Because I don't.
I mean, bioinformation in general, like you get a general idea about what's happening in the field of bioinformatics, and then maybe further you can specialize, like you're doing in Belfast or anyone doing it somewhere else.
So I think rather than it is more important to understanding what you want to do in the analysis per se. Like I am more interested in transcriptome analysis. Okay, for me, the transcriptome is the most important one. So in the multiomics, also in my PhD, I did my whole analysis multiomics at bulk level. Now I'm doing multiomics at single cell level. Some people are interested in drug discovery, like their MD simulation and all. Some people are interested in tool development. Okay, so it is up to you what you want to do. In the bioinformatics, we have different fields.
Okay, thank you.
Okay.
Hello, sir.
Yes.
I have one question.
Yes.
Like UMAP clustering,
Yes.
shall we can we use like some data reported only on cell line, which is already like pure single cell. So does it still need clustering?
Yes. So whenever you're sequencing for the single site monolayer sequencing, you're asking.
Right.
Yes. It requires.
So
Because
still. Okay, and then like for to give an input to UMAP and PCA, what is the input data? Like is the count matrix normalized and then log is the input data, or just the normalized data is the input?
So let me actually, I will actually tell you the exact format we are going to use here. Okay.
Oh, okay, sir.
So
Thank
I
you.
hope, yes. So I just want you people to, if you have, so see, if some packages are not installed using the install.packages R in the R, you can also try the dev tools, and also you can try packages installing by bioconductor. Okay, so I can see some of the people are telling this is not available or in this format. So you can try with different methods like dev tools and then bioconductor. Okay, so guys, if you have installed this three packages, then I would suggest you to go into and download the data. Okay, I think people have already sent the link. Okay, yes. So you can download the data. And let me tell you, yes. So it data has our, okay. So this data has a matrix file, then gene meta files, and then barcodes. Okay. So today we are going to analyze the data in the matrix format, MTX. So I think the girl, she was asking just before that what are the different, what inputs we are going to take. So these are the inputs today we are going to use. So you can get, yeah, you will be using different inputs according to samples like Loom files, SD5, AD files, then RTX is, so we have already processed data. So today we are going to use this matrix, okay? I hope you got an answer.
Yes, sir.
Okay. So have you installed these R packages and everything in your laptop?
Yes, sir.
Okay. So I think quite a few people have installed, so we can proceed further. Okay. Shall I? Okay. Okay. Good. So guys, first thing that you have to just call these, loading these packages. Okay. So here I've loaded dplyr. Here I've loaded dp library password, and then here. Okay. Then one thing that we have to read our data directly from the directory. So you just call this readx function. Okay, some people are still installing. Okay. So guys, you have to give the full path. Okay. Full path of your directory where you install, so like till hg. Okay, you guys want me to say it. So see guys, don't copy. Okay, I can say the code. Okay. Yes, I have sent the code also for this step and for this step. Okay, so please don't copy this path directly because this is my path. Okay, you have to just go to your folder wherever you have this directory. Just right click and go to properties. And like for example, I can show you here. Okay. So for example, I went to the property, then in general, then here you can see the location. And okay, and then I copied and pasted. So people, those who are using Windows, just try to remove this forward classes with the black back classes, because this might create a problem. Okay, just a bit conscious regarding when you're copying. Okay, just remove this forward to black back classes. Okay, so you can see from my screen, the path should be defined here like this. Okay, otherwise you will get the error of the directory that you are not not loading the right directory. Are you guys able to proceed with this step? Loading the data into directory, calling the data from directory. Okay, enable the matrix. Open. You don't need to open the matrix, man. Science re, just give the path. Okay, path of the folder where this matrix and gene and barcodes are present. Just give the full path. Here, like here, I have it also. If anyone has not able to done that, just please let me know. Path to folder, not matrix file. Kajal, path to folder, not matrix file. Okay, so this is like HG9 folder is there. So you have to give the full path to HG9 folder. The one thing also you can do, like you can set the paths directly from the R. So you can actually get to know where the different, what is your working directory. Then you can set directly from here. So just copy paste that path here, and then you don't need to define the full path. Okay, then you can call it. But this is up to you how you want to use it. You can just define in the R or just define in this read 10x function. Please let me know if anyone is not able to call this function. No, we don't need to import the gene TSB. You have to just define the path. So see what happened. If you will read the directly, define this directory to this read 10x function, it will automatically get the gene barcodes and the matrix and gene TSB file. You don't need to call it individually. That's why the reason I'm asking you to restore your PBMC, all the 10x, this files in the one object, pbmc.data. So if you will just define the path of the directory, then you don't have to call it individually all these data files. Could not find. Okay, so if you are getting error read 10x function not find, this means that you have not called the Serat library. You first install the Serat and patchwork in the DPL by R. So if you have not installed, then how this possible? Read 10x. If you have installed, then how it is possible you are getting this error? Couldn't find function. Oh, so you are using a small x. In the read 10x, you have to define the. Yes, I have already provided Karambir. You can get it here. Copy. Okay, you can. Oh my God, this is too. I have to check it over. Yes, this is it. So, Sangamitra, yes. So, Sangamitra, you're calling that 10x wrongly. You have to define read 10x capital X. So, yes, now you're getting the data directly not found. This means that you are providing the wrong path. Could you please say a path? Because you are providing the wrong path then. Yes. Data direct not found. It is a code.
Hi, Anush. This is Shashanga. I've provided because I've used tab line, so I don't think the path is wrong. But is my line the code line? Is it correct? I'm using, and I'm also using in Mac OS platform.
Okay, no issues. So, could you just paste the exact code,
once more,
exact code you're using right now?
I'll just pasting my code.
No. This equal to sign is not correct.
Which one?
The data.dir, then you have this is something minus. You have to make it equal to.
Okay, maybe.
Okay, this one.
Let me, yeah, sure. I think. Okay, yeah, thank you. It's working. Thank you very much. Yeah.
Okay. So have you shared the code? I have shared the code. Is there anyone who is not able to do that? Guys, please let me know. We can, because this is interactive session, please. Could you please tell me I think Raksika.
Sir, can you show me how to open from R file? From file open.
What open? Packages are not installed. Okay. So Deepthi, what is the problem you're facing while installing the packages? Could you try some other methods like I told you, the dev tools and then bioconductor? Yes, it is showing not available in some of the default version. You can try the dev tools and then bioconductor. And the Sirat package loading. Okay. So Survi, could you please say the error so we can rectify if I can with the limited time?
Out of these three files, what we should do, sir? Genes.tsv or barcodes tsv?
Yes.
What?
So all these files we are going to use in the next step when we will be creating this Sirat object. Okay. So once this PMC data is fine, then we can proceed further. So I think we can proceed further. Okay. So first thing that once you define your TSP file, gene TSP, then matrix, then barcode file, then first thing is that creating the Sirat object. So guys, creating the Sirat object is very first and most important step. Okay. So from here, your all analysis, then all steps, then all the outputs are going to save into this object only. Sirat, and this is an interactive object. Okay. So from whenever at any point of time, at any step, you can just check what are the different modalities installed, what are the different analysis installed, what are the different I would say different type of differential expressions and markers are installed. So everything will be captured and stored into this Sirat object. I would say barcode TSP required, but okay. So we just, you don't need to do any. I think you have to just decompress the file you have downloaded using the link I shared with you. Okay, let me again share this folder. Okay. Okay, so guys, if you see this matrix is present. Okay. This filter basic matrix and then filter by items, they have HG9 folder. And in this HG9 folder, you have this one. I don't know how it is possible. Yes. These three files only, nothing like nothing. will tell me that I know these are some people getting errors. So is there anyone who has done that? So I can take that if someone is running with me. So we can match our output with each other. Okay.
I know this choice of minimum cell, like minimum features, is it kind of a standard thing to take like 3 and 200?
It is just like default one, but you can tweak this parameter according to your biological questionnaire according to your requirement. But this is some default parameters I'm saying. 3.
And so like if you have like a knowledge of like what you are using, then and you
so
can
see, if we have if you want to improve the number of quality and you don't want to take the cells which is having the poor quality cells, like improve the quality, so you can take you can increase the minimum number of cells and the feature also. Okay, so if you will just if you will increase these numbers, then you will get less cell. Okay, if you will just reduce the number, you will get more cell population. But the thing is that handling the quality population would be much easier than handling the poor quality cells.
So like increasing the minimum number of cells. So if I got it correctly, if I increase the minimum number of cells, that means it will be a better quality. That's
yes. So it would be the genes, like 200 genes should be present at least in 3 cells.
Okay, 200 genes. Okay, that means 3 cells in part.
Yes. So if you increase the cell numbers, then it will reduce the cell number obviously, because you are reducing the number of cells. Okay.
Okay.
Okay. So guys, I think I can see a couple of people have installed and going at the pace. So meanwhile, those people have getting error, just try to resolve the errors, but we can go and come back to you. Okay. So in the first step, we are just running. We have run. Now you can see we have restored our cell matrix and gene barcodes and gene specified in this Seurat object. And you can see this Seurat is we are having the 13,714 features. Feature means gene here, guys. Okay. Across the 2,700 samples. So 2,700 is the cell numbers. Okay. So we can also check number of cells. Yes. So right now in this object, we have stored 13,714 genes and 2,700 cells. Okay. Do you also getting the same thing or not? Some of you can please respond so I can get confirmed that yes. Good. So first thing that we have to label the mitochondrial population present in our cell types. Okay. So first, to map our mitochondrial population, we have to define this percentage.empty column in your matrix. Then if we have just added, then let me show you the data. Okay. So do you see we have the different cell, okay, present here, different like same origin ident, then the N count, then N features, and the percentage mitochondria. So do you see the when you are checking this heading PBMC at the metadata using this command? I can tell you. Good. Okay, so do you see such an output? Okay, good. So very nice. So once you have the first QC plot we are going to generate here is for the features and for the count and for the mitochondria. Okay, so you can see this is the plot I presented you people also. Okay, so you can see our data looks fine because it is the number of features and number of counts and number of the percentage of mitochondria should be lower, because the more the number of mitochondria percentage mitochondria cells having, the more dead cell population you expect in the data. So Salin is asking what this code do. I'm getting error. Error used without the Shraddha, the problem with the path, I think you have to define it more clear way. Yes, Salin, for your answer, this query that here, Salin, we identify the percentage of features, like we have certain genes we expect should be present in the mitochondria. Okay, so we already have this set of genes, so we try to check what are the expression of those genes in my all the cell types, and then calculate this percentage mitochondria. Okay, so once you have the percentage mitochondria, then you can really get to know what are the percentage of mitochondria present in your cell. Then second thing, if you get to know the cell percentage, then you can really filter the cells. The more number of cells having the higher percentage of mitochondria, that means they are dead cell.
Okay.
So sorry for your answer is that this is the
Yeah.
plot we are doing.
Yeah, thank you.
Okay. I'm getting warning message. You can ignore bonding. Yes, I'm very well replied. Okay. I think some of you are really doing great. That's good. We can proceed further. So this is the first QC plot, guys. Okay, we get. I hope some of you have get this plot. This QC plot. I can see Sangha is there.
I am getting this, like I'm getting only the first of the two, like count and this, like.
Yeah, sir. So basically, Sangha, what's am I pronouncing correct, Sangha?
Yeah, Sangha.
Yeah. Oh, okay. Sangha, you might not be running this one. Okay.
I'm running this one, like VLN plot, PMS features.
No. You before VLN plot, you have to run
maybe
this.
I missed that one. That's what you're saying. Okay.
Yes.
So please, this is basically to identify the mitochondrial cell. This like that.
Yes. So this will actually identify the mitochondrial percentage in all the cell types.
Okay. 1 minute. Yeah, I'll just go it. Yeah.
Okay, so it's working. Okay. So I am is asking about, could you please explain the accuracy matrix? Yes, sure, I am. I can. So guys, if you can see the first plot and feature RNA plot, okay. So ideally, the quality should have at least optimal number of unique genes present in all each cell types. Okay, but if you are getting the very low number of cells, lower number here, and the very high number. So if we're getting high number here, this shows that there might be doublet of cells in sequencing. Okay, the ideal case should be you should get the optimal number of features, uniquely present features in all the cell types. Getting more high number, this shows the poor quality, you might have the doublet of cells. Okay, first thing, if you're getting the lower number is very high, this means that you should have your cell is not having the sufficient number of genes. Yes, please. Okay, this is the similar case with the count number also. It should be the optimal, having the lower number and not very too much. In the percentage, mitochondria should be expected a lower side, because the higher the percentage of mitochondria, you expect the more dead cell.
In these two graphs where you are saying that both should be on the lower end, so one is for 1,000, the horizontal axis has 1,000, the other one has 5,000.
Yes.
So in that case, that other one is a bit higher than the previous, right? It is not 1,000. So
Yes. This is scale should be scale is you're correctly said. Okay, so this like overall scale, this should be in the lower side, I think.
what does this represent? Like, as in, like, your number of cells each cell have a particular RNA present, or what is the?
So this shows that this is the transcripts number.
Okay, so each cell, how many transcripts are present?
Yes. The transcripts, having the more number of transcripts might be the problem. Okay. 5,000 is something optimal number.
Okay. And what is this feature thing? Like, feature means as in your property.
Feature means the gene.
Okay.
So suppose you get the gene, okay, and then for the gene you get the transcript. So basically on the number of transcripts define the genes.
Okay. So you are saying that this n count RNA should be higher than your n feature RNA always, because there are genes that are getting transcribed, right?
Yes. So it is expected that your genes would have more transcripts, because the multiple transcripts will comprise one gene. The expression of genes. Okay.
Yeah. Okay. Got it.
Okay, guys. So please share the code. So Vinayak, what code you are asking? Could you please tell? Because I have already shared a lot of codes. Okay, so I think QC, this is the first level QC. Okay, then second level, that you can check the correlation between these two QC, features count and then percentage mitochondria. Okay, this should be the problem. Yes. So can you please? Yes, as I will do. I will do. Yes. So Vinayak, you have to just run these two commands. Okay. The feature scatter for the count versus percentage mitochondria, and the features count versus the features and count versus the end features. Okay. So let me explain you this plot, features versus count. Okay. So if you have the cells, which is like cells which is having the higher number of features, might be, but the less the number of transcripts, then these cells are not good to analyze further. Then in other case, you have the cells which is having the high number of transcripts but the lower number of genes. Okay, then this is also not the ideal case. Okay, the ideal case should be that you have the equal proportion of the, like not equally, like the optimal proportion of the transcripts and genes present in your cell. Okay, so that's why the genes present in this straight line having the better good correlation between gene features and the count should be taken for the further analysis. So in our case, we can call it that our data is a good quality data, because we are getting around 0.95 correlation between this feature and count. Okay, so we are not getting any such case where these are outliers. Okay, so if we would get any such cases, so we could have removed it, these outliers coming here or there. Okay, then
Sir, what are those 2 points in the top right hand side? There were these two. Yeah.
These two.
Yeah.
So this shows that there are the few cells which has the high number of transcripts and high number of genes also.
Okay. So
okay,
most of them have.
but it is not a problematic here. Okay, because cell have the, suppose a cell cannot have the more number of transcripts and the less number of genes. This will show the discrepancy in the sequencing. Okay, so for example, if you have a cell for which the cell is having for a gene A, it is having ample amount of transcripts, and for others it is not having any transcripts. It means that the cell was selectively sequenced for one gene only.
Okay.
Okay, so for that this correlation should be high. For valent blood equals to variables. Aishwarya, you have not read the file correctly. That's why you're getting such errors. So shall I proceed, guys? I think if someone of you is like you have achieved. Are you able to generate these plots? Okay, Sangha, good. So we can proceed, okay? Yes, good. So, like guys, see, we are getting good. So we were getting these plots. On the basis of these plots, we are now subsetting the data good quality data. Okay, although we have good quality, we are just defining the parameters. Okay, so here again we are subsetting our data like, and features our cells should have number at least more than 200 features. Okay, and less than 2500. And the percentage of mitochondria should be less than 5%. So we are taking the cells which is having more than 200 genes but less than 2500 genes. Okay, and also the cells should have less than 5% mitochondria. Okay, so this shows that we are going to use only the cell types which is having such characteristics. First thing. The second thing, that subsetting this would define that we are taking only quality cells for further analysis, because this step is something you are going to use this subset for the further analysis. So if you won't apply such any subset of the good quality cells, then you might see any you might get error, and you might see problems in the lower NRC or downstream analysis. So it is better to be sure with your matrix, the count quality analysis, and then we can proceed further. Okay, so here we have subset. Then first step is the normalization. In the normalization steps, this is the first time you are going to normalize your data. Okay, for normalization we have different scale factors and the normalization method also. So you can optimize this normalization method according to your requirement. Sometimes you need to optimize it according to the data which you are analyzing. Might be a lot of discrepancy and a lot of noise present in the data, so you have to try some other methods also, but in the default condition we use the log normalization. Okay, so here you can see the normalization is done. Then identifying the variable features, the features which are variables and which are differentiating the cell types with each other. Okay, so most of the cases we like, since in our case we were having around 17,000 features in our data. Okay, so what is the optimum normalization scale? Okay, so scale factor 10,000 is the default and the optimum one, but you can tweak with the scale according to if you're not getting your downstream good. So this is default and scale 10,000 is fine, optimum one. But you have to tweak when your downstream is affecting, not in the line. Okay, so for downstream like analysis, your cells are not clustering, okay, on the basis of the cell types. First thing is that. Second thing that you are not getting the optimal differential expression which is expected between all the cell types. So clustering and the differential expression would be affected if you won't do the normalization properly. Okay, so have you guys done with the normalization? Yes, good guys, please, I want you people don't miss any steps. Please responsive, please be active on at least in the chat box. So I know it's virtual, but at least it would be very nice if you can just keep me informing in the chat box. Yeah, that's great. Good. So Gunapati, I can say the whole script after this session, and this session is recorded, so you can go also check the session, because right now I'm not copy pasting the whole script because it might lead to problematic for the others participants also. I will share the full scripts.
Okay, can you please at least share the code? Like once you like ask us to run the code, can you just copy paste that specific?
Okay, sure. I will do that.
Yes,
so
please. Thank you.
For the normalization step, okay, I'm doing that. Okay, guys. At least. Okay, so Jared, these scripts steps are in the chronological order only. Cancer is asking if there any documentation so we can choose the value which we are putting in the parameters. Yes, I will share some of the tutorials where you can get to know exactly how these parameters can be tweaked. Okay, what is the significance of FFS? All the zeros is the gene count and 9 all the zeros in the gene count. Arthik, could you please explain your question? Anyway, okay, we can proceed. You can ask. Okay, Rakshika. That's not a problem at all. You are running R for the first time. No need to worry because you might get some errors, but you can optimize it. That's not this is not a problem at all. No, Arjun, I haven't heard you. Could you please explain it again? I'm not able to hear. Okay, no issues. Okay, we can go. So guys, and the next step after normalizing the data, we are identifying the variable features. So we are taking only 2000 genes for our further analysis. Okay, so
can you hear me now? Sorry.
Yes, Arti. Yes.
Oh yeah, sorry. So what I was asking was, we are doing a log normalization, right? So if you
Yes.
have zeros in your matrix, log of zero is going to give you like errors. So how are these handled?
Okay, so yes, you have rightly said. So it has replaced with the NA.
So you do log of NA, is it?
Yes. So no. So suppose if you go do the log normalization of 0, okay, then you won't get the any. You get the infinite or some error.
Yeah,
You
you
won't
get
get
minus
the,
infinity.
yes. So there is nothing like less than 0. So you won't get minus when the value is less than 0 for any log. Okay.
Yeah, I understand that. So if you're having a gene matrix and you have zeros in that, so when this normalized data is used,
So it is already yes. So it is already handled. So you won't get any such errors because while developing this software, people have realized this and they have rectified this error. You won't get, if your matrix is having 0, you won't get any error.
So is it removed or is it?
It is
made to
removed.
one?
It is rectified.
Meaning?
Rectified means you won't get such error. Like if log
okay.
normalization standing 0 is there, then definitely if you do the Excel, in Excel you do the 0 of log normalization, then you will get error. Okay. Okay, so it would remove any such cases. It is already taken care when it was doing the log normalization steps.
Okay, yeah, thanks.
Sorry, did you hear me? Then could you please describe the nature of data in the files, how it is structured, just to get an idea. Okay, Salin, we can do that. I will explain you this, all these modalities, and the analysis will be stored in one, our Sirat object. Okay, that can be done. So I think, have you done this? Have you guys run this find variable features? I have shared the script with you already. Okay, then after finding variable features, let me show you some top 10 genes. Okay, which we have selected from our this when we identify the top features. Okay, so see, out of 17,000, there was a 11,700 which were not variable features. Then we identified only 2,000 genes, and then we can also plot them. For the first time, what to do after installing, or should I do command I need to type? So, okay, let me show you those scripts. Okay. Okay, so you can actually plot your variable features, which we have selected around 2000 variable features. And you can also label the top variable features we are getting in our data. Okay, so like here you can see some of the features, PF4, GN, G11. So then next step is that we have to scale our data. So we have selected all the genes and we are scaling our data on the basis of genes present in our variable features we have selected right now. Then we will do the dimensionality reaction, just one dimensional reaction, like PCA is the first dimensional reaction we are going to use right now. Then in the PCA, you see the different components we are getting. Okay, and for the different components, we are getting the positive and negative genes. And then if you see the top positive and negative genes, like top 5, then you can also get to see what are the different genes are there, how the expression is changed. Then we can do the plots. Okay, someone was asking why we are using PCA. Okay, and then why are we using UMAP? Okay, so here, can you please hold on? Yes, I will. Sure. Okay, let me see. So Stalin, just try to run the label points using the command I'm doing right now. I'm giving you. Okay, here. And now you just have to plot. No. You have to plot just this one. Okay. So like here you can see this is, and here you see we are getting the plot.
Yeah, actually, I'm getting the plot, but only labeling that points. Label points is not working.
Okay. So label point is not working. Then there might be, the reasons could be that the top 10 where we are storing this label might be the problem. Okay. So there is nothing to worry.
I'm also getting an error that the label point, it can't find the function. I'll just paste the error message.
Okay, you can ignore this error for a point at what timing. It is not going to affect your downstream. Okay. Yes. We can ignore. We can afford to ignore this error. Can you please scroll a bit after setting directory for? Okay, Priya, I can. Yes. So Priya, could you please? We are on the line. This is what you want to see. Yes, Manjusa, I can see this error. So you can ignore this error for timing. We can solve it, but okay, Priya. Okay. So guys, could you please let me know that how many of you have plotted this team plot for the PCA? Yeah, any of you want me to slow down, because yes, I know some of people. Okay, this one are good. But any of you who has come to this step and could able to generate this team plot for the PCA, apart from the Priya and Devasmita? Done till label points. Okay, Kajal. Okay, done till label points. Okay, just do the scaling, Kajal. We can wait for you. Do the scaling, run PCA. We can wait. Can you share the screen? Yes, sure, Shweta, sure. I can share. Guys, those who are worrying about the script and everything, I will share the IDS file, I will share the script, I will share the recording and everything. So no need to worry about the resources. Just for this, just right now focus on the analysis. No need to worry about resources. I can share even additional resources for you to people to understand the single cell analysis in a better way, and some good tutorial also apart from this script. So no need to worry about the script and recording of the session. I will share it with everyone. Okay. So Salin is asking what is the PC1 and PC2. Okay. So Salin, PC1, PC2 and all the components which have the different cell types present in those components. Okay. Then every component is different from each other because it has the different set of genes and expression. Okay, so if you see for the different components, we have different genes present in those components, and those some of them which are positively regulated and some of them negatively regulated. Okay, for example, for PC1, this is the top. If we take the top 5 features, then if you CSP3, Hang on, who is that? Okay, so silent PC 1 and 2 and all these PCs are the different components where the different genes are clusters. Okay.
Yeah,
Okay.
okay. So finally, we are plotting between PC1 and PC2 only.
So silent, basically, this is just representation. Okay. So when we will be doing further dimensional reduction, we will be using all the components like 1:15 components. Okay. So that's why the reason we are doing the UMAP something. UMAP like analysis where we can plot all these components in just one 2D graph. Okay. So in the 2D graph, it is not possible to plot more than 2 components. Okay. Because there's a lot of components you can't plot in the 2D graph. But when we'll be doing the UMAP, so we would be able to get more precise clustering, because that's why the UMAP is required.
Oh, thank you.
Yes. So guys, could you please let me know, shall we proceed further? Because I'm waiting for you people. Yes, Kadal said yes. Okay. So, okay, Sangha, good. So we can also in the, sir, can you please explain how the normalization dimension parameters changes with the trend data types? Okay. So dimension reduction parameters. Okay, Aishwarya, I can explain you this dimensional reduction and normalization while by tweaking the parameter within this data set only. Okay, so I will show you that if I will tweak one parameters, then you see how this impact is impacting the cell clustering directly. Okay, so I will show you the impact of these normalization and dimensional reduction changes in the different data. So right now in this data only, okay. I will do that. Okay, so once you have this PCA already done, then you can check the plot the different like cell genes present in your components. So you can see this is the same. Okay, where we are getting the top genes for the PC1, which is the positive and with the negative, how these are clusters. Okay, so it is just to get an overview, what are the different cell, what are the different genes are present in components. Then it is just nothing like for the you since we have taken 15 components. So the guy who was asking, I think Shalini, yeah. So Shalini, you can see we can also plot the different components individually. You can, I can zoom it so you can actually get to see it better way. Yes. Okay, so rather than just going one by one, you can also plot the genes present in all the component and how their expression is changing. Okay, so if you might see like PC1, PC2, PC3 till PC3, we are getting for PC1, PC2, we are getting very differentiated and very clear clustering of the genes. From the PC3, we are getting some of the genes clusters, but not the other one. And PC4 also some not clustering very properly. Okay, so this is how you can see. This is in the normal PC also you can see most of the components genes are accommodated in component 1 to 3. Okay, and then later on all the components having the different genes, but not the very specific features. Okay, so this is just, guys, this is all these analysis. This is just a part of visualization to get to know about your data more better way. Okay, so here, once you know what are the different components having the different cells, having the different genes present in those components, then we can actually calculate the optimum number of PCs should be considered for further analysis. Okay, so we are calculating this Jack straw. So I can also copy paste this to everyone. So Divakar is asking, you are suggesting that UMAP is better than PCA for first. Okay, so yes, UMAP is better for the clustering as compared to PCA. Yes, that's why the reason we are using UMAP. I mean, Tisni is another algorithm similar to UMAP, but UMAP has outperformed Tisni. So now Tisni was the first developed, then UMAP came, but now UMAP has better clustering than Tisni. So this is all the clustering, clustering algorithms like PCA, Tisni, and UMAP. UMAP has outperformed PCA and this Tisni both. So if you see the older paper, most of the people were using Tisni for the clustering. Now you will see most of the paper with the UMAP. But still, I won't comment that it is up to you. If you're getting better clustering with the Tisni, you can go with the Tisni. But yes, most of the people are using now UMAP. Okay. Okay, so someone is asking why are we doing this Jack straw plot? Okay, so basically Jack straw plot will provide us a visualization of comparing all the distribution of p-values for all the PCs, okay, with a uniform distribution. And this will tell us that how significant is that particular PC component. Okay, so for each PC component we will get a p-value, and on the basis of that p-value, those who are not significant, we can remove those PC from further analysis. Okay, so while doing the UMAP, we can have a choice to select the PC components which are statistically significant. Okay, so let me plot the Jackstraw plot for you so you can actually get to see how this look like. Okay. Okay, so if you see our sum of the PC as expected has a very high statistical significance, like PC 1, 2, 3, 4 until 1, 2, 3, 4, 5, 6. Okay, yes, somehow 6. Yes, so we have very high p value for these PC components. Okay, and some of the components, like you can see, those are not statistically significant, like PC 14 and PC 15. Okay, so this will tell us when we'll be selecting the DIMS for the further analysis in the UMAP. Okay, so what are the up to which we should take the components for the further analysis. Okay, that's why this Jackstraw give you better understanding of what are the component which is significant to be considered for the further clustering. Okay, I hope I could answer your question. What does DIMS depends? Okay, DIMS represent the components, how many components you want to take. Like DIMS 1:15 means you are taking 1 to 15 components. DIMS 1:10 means you are taking 1:10 components. Okay, Manjusa, you can ignore such errors. Yes, it may be errors because of sometimes your visualization window is too small to visualize such plots. Okay, Deepthi, I hope I could answer your question. What DIMS represent? Similar, like Jackstraw, you can also use this elbow plot. You can get most of the PC component which are showing the higher standard deviation. So see, Jackstraw plot and this elbow plot just selecting these components for the further analysis. Okay, so like in our case, I can see that till the PC 10 we are getting the genes which are actually having the high standard deviation. Then up lower than after PC 10, the genes components are not having very high standard deviation. So in optimum cases, in my cases here in this data, we can take the DIMS from 1 to 10. So like taking the components 1 to 10 would be fine for us for further analysis. So that's the reason we are taking DIMS 1 to 10 for finding the neighbors and the clusters for further analysis. Okay. So guys, you can see, you can decide on the basis of this elbow plot and the Jackstraw plot we plotted. Okay, how many components you want to take. Okay, so here I have decided to take 1 to 10, like 1 to 10 component. And then you run this neighbors, then you run this resolution. Okay. Then you can actually run the UMAP. Okay. And then you can plot it. Okay, so here you can see we are getting 9 clusters. And someone was asking if I will tweak these components and the resolution, how this will affect my whole analysis. So let me show you the example. Okay, here. Okay, let's do one thing. Let's play with the parameter right now. This will change. Okay. So for example, here I'm increasing my dims from 10 to 20. Let us store it to one new object, PC1. Then taking this PC1 and suppose taking the resolution 9. Okay. Then same PC1, then again take the dims 20. Okay. And then let's see how this clustering looks like. Okay. So here I am using the 20 components, then resolution is 9, and then we are getting different number of clusters. So you can see the okay, and then so you can see the clustering has been changed. So this is the new clusters which look like, and this is the older clusters. Here cells are more condensed as compared to these clusters. Okay. So this is how you're changing with the parameters, dims and resolution can change your clustering. Sometimes it can also change the numbers. So let me try with the new. Okay, let's do the 2. Okay, guys. So like here, when I just changed my dims and resolution, the clustering changed, but the number of clusters was similar, like 9 clusters, and here also 9 clusters. The only thing changed was the cells were more condensed when I increased the dims and the resolution. But when I reduce the resolution and taking the same components, if you see, the number of clusters has been reduced from 9 to 7 only. So this is how you can see the change with the tweaking with these dims and resolution may change your clustering. One thing I can also show you people for this analysis. This will be good to know. Okay, so we are using another algorithm. I was telling you people to, you can look into that, like Disney. Okay, so we are also clustering same sample with the Disney. It will take some time. Then while plotting our object, we can change direction from UMAP to Disney. And then let's see. Okay, so if you see in the case of Disney, as I told you, the clusters are not, cells are not more condensed into clusters. So same plot in the Disney, you can see cells are more dispersed. Same plot in the UMAP, cells are more condensed. And okay. So what exactly my resolution? Okay, so Prakriti resolution means increasing the cell condition. Okay, so here in case if you increase the more resolution, then cell will be more condensed. Shall we proceed? Guys, are you able to run through this step or not? Let me take. Computing nearest graph error in validity method algorithm plus matrix did not found. Silent, you might have missed the previous step, I think.
I could plot till just before the command, everything was running smooth. From the find neighbors command only, this error came up.
Find neighbors, okay, this is coming, okay.
Elbow plot, till elbow plot, it was running fine. Just from the PBMC, find neighbors, PBMC dims 1 to 10. That throwed up there.
This is pause. Okay, you are using the same command, find neighbors, PBMC. Minus 10. How is this possible? Okay, let me check. The region. Okay. So this is error due to some other package coupled with Serat is like matrix. So this is the problem just because of matrix. So you might try by, you may rectify this error by reinstalling the matrix.
Okay.
Yes. So matrix is other package coupled with this Serat. Okay, so this is error might just because of matrix. So you can rectify this.
Package, reinstalling matrix package.
Yes. Matrix package could help you.
Could you tell me the package name exactly? Is it
Okay,
like
sure. I can tell you. Matrix. Getting error. This is step PBMC. I have sent this matrix. Okay, Barlow. So this might be. Did you install the Serat today? Hello, calm
Yes,
down.
today only.
Yes, yes. So this is might be the little bit the new version. So some people have reported this bug. People have reported this bug. Yes, yes. It can be done. So yeah, as of now, you can just follow this what we are doing, and you can rectify this error by installing the previous version of Serat. Because this is something which problem with the I think newly installed Serat. People are reporting this bug. So guys, shall we proceed further? Could you please let me know so we can actually move ahead? Yes. Okay, Sangha, thank you. Thank you. So the next step is like in analysis, you have these different clusters. Okay, so you know that your cells are clustering into different. So your cells are clustering differently. But the next job is that to define these clusters. Okay, so like what are these cell types? What are these cell types? What are these cell types projected in your data? Okay, the next job is very crucial because cluster annotation is very important. You know the cells are clustering differently, but you know what are the cell types to be called. Okay, so first thing is that you can identify the markers. And how to identify the markers? You have to identify the differentially expressed genes between your clusters. Okay, so for example, in case of our default here, you can see, yeah, this one. So we got the 9 clusters. Okay, so we can calculate one thing. We can first do the identification of differentially expressed genes for this, for example, for clusters 2 markers. Okay, so here what we are doing, we are comparing cluster 1 versus 2. Okay, okay, so we had to identify, and you can see. So these are the top genes which are highly expressed in cluster 2. Okay, so on the basis of these marker genes, we can label clusters, we can label and we can assign a specific cell type to cluster 2. Okay, similar for the cluster, other clusters. So like here, we are trying to identify the markers for cluster 5, which is different from cluster 0 and 3. Okay, so here you can see the different clusters. So this is all about the cluster optimization. Plus, how do you identify the relevant marker for your clusters? Okay, so these markers will be decided on the basis of expression of the genes. Manjusa, you can proceed further as of now. No need to stuck with this Jackstraw plot. Just try to run next steps just to see. Okay. So one thing is that you identify the different markers for all these clusters. Second thing that you can identify markers for all the clusters. Okay. So you can really compare one versus all. Okay, so here we will be identifying the markers one versus all the markers, differential expression for all the clusters. Okay, so here you can see this is calculating clusters, different expressions for cluster 0, 1, 2, 3, 4, 5, 6, 7, 8, because since we have the 9 clusters in our UMAP data. Okay, so once we have the differential expressed genes for each clusters, you can get to know what are the different genes and what clusters they are highly expressed. Okay, so you can get to know the top differentially expressed genes for each cluster. Okay, and for example, we have some genes. If you want to know the expression of some marker genes in the different clusters. So for example, this MS4A1 and CD79A. So you can get to know where these genes are highly expressed. Okay, so you can see these two genes are highly expressed in cluster 3. Okay, so this is how you plot your marker genes and get to know what are the different cluster in which the clusters these genes are present and they expressed. Okay, similarly you can also plot some more marker genes for the clusters like these genes like NKG7 and PF4. So you can see this PF4 is highly expressed in case of cluster 8, and then NKGN is highly expressed in case of 4, and also more like highly expressed, but here in case of 6 also, and cluster lower, not very high, but moderately expressed in case of cluster 5 and 7. Okay. So, and in addition to that, rather than plotting these individual genes, you can plot the multiple genes and check how these genes are expressed in different clusters. Okay, so if you remember this plot, okay, yes. So this was our original UMAP plot. You see the 9 clusters we have here. Okay, and in the same these settings, you want to know what are the different marker genes are expressed in different clusters. Okay, so just remember this clusters, and you can see how the different genes are expressed in different clusters. Yes, here. Okay, so you can see some of the genes MS4 even specifically highly expressed in these clusters, some of the genes expressed in this cluster, some of the genes expressed in all together in these clusters, in the both clusters. Okay, so this is how you can see this. Oh, great, Sarin, great, good effort. Yes, this is the way we need to learn. Good. And apart from this, plotting these different clusters, you can actually plot all the marker genes, top 10 marker genes for each clusters. Okay. I can do it. Yes, so here you can see the top 10 genes for each clusters, okay, and the expression profile. So on the basis of the expression of the genes, you can also define the clusters, okay. So if you know that this is the set of the genes which is, marker for a particular cell type, then you can label clusters according to expression of those genes. Then cluster 1 according to that, then cluster 2, then cluster 3, then cluster 4. Second thing is that if you think that two clusters having the same set of genes, then you can also label two clusters with the same name. Okay, suppose for example, if I think cluster 4 and 5 are the same cluster of same cell types, so I can label the cluster 4 and 5 as a like neuron cell. Okay, if I think that these three clusters like cluster 1, 2, 3 are from same are the cluster of same cell types, like for the glial cell, so you can name all these three clusters as a glial cell. Okay, so it is up to you. Okay, so what are the markers genes expressed? Okay, if you think that more number of clusters could be named as a one cell type, you can do that. But you have to be very precise and you have to be very detailed and comprehensive while deciding the cluster's name, because this is something on the which we are going to build your hypothesis, which we are going to build your whole downstream analysis. Okay, sir, it is still showing the error after doing the next line of code. Jackstraplot as a validated. Manjusa, you can ignore plotting this jackstraw plot as of now. And could we proceed?
Sir, for elbow plot also it's showing the same.
Okay, so there might be in bad. So how are you running this code? Like in RStudio?
Yeah.
Then how is it possible? The graphic state, you're showing the invalid graphic state. Let me check. Why are you getting this error? It's 250. 250. Manjusa, could you please try with the lower number of dims, like 1:10? Okay, guys.
No, sir, it's showing the same.
Okay. Also, there might be some problem. This can be the problem. There might be problem with some components, I think. Names. Invalid gap. This is something not related to your code. So you can move further. This is not going to affect your analysis because this is just a visualization problem. Anjusa.
Okay, sir.
Yes, yes. This is just a visualization problem, not a problem at all. Okay. So guys, are you sure? Because we are now going to annotate our clusters. So till now, is everything clear? I'm expecting your response in chat box. Can we proceed further or not?
From this plot, cluster 0 and 2 are looking similar.
Yes, yes, yes. So we can name them. Anyone else? Please, guys, please be interactive. It's very difficult to interact in virtual, but we are trying our best. So please let me know. Can we proceed further? Yes, Sangha saying yes. You know, the people there.
Yes, sir.
Okay, just Kajal is saying just a minute. Okay, we can wait for Kajal. No issues. I can do one thing. I can paste all these commands again. Chat
So when
box.
is the break?
So you need a break? Yes, yes.
Yeah.
We can take break, no issues. We can take 5-minute break, guys, okay? Then let's come at 11:45. So, hi everyone. I hope this work is 5-minute work is now 10-minute break. Are you guys back? Or just still on the break? Oh my God, so many people left. Celine is asking, please enable copying from channel. Celine, this, I don't have that right to do that, I think. Let me check if I can. No. Okay, Devasmita Paul is saying yes. Okay. I'm just waiting for more confirmation to proceed further, because since guys, this is the last step, we are going to do that. The labeling our clusters. So this is very important steps. Okay, few more. Sangha, Helen, good. Okay. Okay, Julie is also good. If you guys could just, simply type yes in the comment box, it means good to know that you are active in that. Okay, good. Lot of yes. My God, thank you. Okay, I thought you guys left. Okay. Got great. I'm happy to see lot of people responding now. Okay. Silent, this is not, I don't have any such right to do that, I think. Enabling.
It shows like by default copying from the chat is disabled. So I'm not able.
Yeah, that might be the host who has created the link.
The host has to enable that.
Yeah. So guys, don't worry. I don't know where to get this. How will I get the contact detail of you guys? So I can email these scripts and recording to all of you, because this is very useful.
Yes, sir. Thank you.
So no worries. I will email the scripts and everything to you people. Don't worry. Okay. Don't worry. So see, look, on the basis of expression of the marker genes, we decided to assign the clusters. Okay, and this cluster should be in the order of your clusters. So like if you want to name your cluster 0 as a naive CD4 8 cell, okay, so it should be in the order of your clusters you want to. Okay, it should be not random. Okay, suppose name CD4 for the 0, cluster 0, CD14, CD 14 plus mono for the cluster 1. Second thing also, that if you want to like have the similar name for the two clusters, like the same name, then you have to retype it again. Okay, so for example, if I want to name cluster 1 and 2 with the same cell types, then I have to write CD14 plus mono, then again CD14 plus mono. So just the basic thing to remember, that if you have the 9 clusters, then you must have the vector size should be 9. Okay, here the cluster ID vector should have the 9 names in there. Okay, it may be duplicated, that doesn't matter, but if we'll supply the less than the 9, then it might be a problem. That okay, so you always remember the number of clusters. So the number of clusters should be always matched with the number of clusters IDs. Okay, so if you see here, we have stored our cluster IDs in the cluster, and you can see the number is 9. Okay, then we have to just add these IDs as a label to our select object, then rename idents, because our default idents should be, I think, our default names are now, I think, the clusters, the Sirat clusters. So that's why we are naming these idents to as cluster IDs now. And you see our clusters are named. Okay, so we have the different clusters now. This is specific, this cluster having the space pixel, the FGR3A plus mono, and this cluster is specific DC1. Second thing is that if you see the clusters are, the clusters, cell types cluster in different samples have the distinct clusters. Okay, so like suppose pallets not present anywhere else. Okay, this NK cell types are present only here. So if you see the blue dot present here, then this might be a poor clustering. Okay, if you will see these, DC, like some magenta colored dots are present somewhere else, then this will show the poor clustering. If you are seeing the similar cell types present together, not present here and there, then this shows a good clustering. And good clustering means good downstream analysis. You can proceed further for any set of analysis from this data. Okay. So how I determine which cluster, what cluster is which? Okay, so Rasika, Ravindran, Rakshika. Okay, so I told you that to determine the which cluster is which, for that you have to check what are the differentially expressed genes present in these clusters. So for example, for cluster 1, you are getting the markers for a particular cell type is highly expressed, then you have to rename and assign that cluster according to the expression of that marker genes. Okay, so I have already explained to you, or we decide on the basis of the name of the clusters, on the basis of expression of the marker genes. Yes, the marker gene is based on literature. Yes, and from the previous observation. Yes, the marker genes are based on the literature and previous observation. We know now, like for example, AQP4 gene cannot find UMAP in the Sirat object. Okay, so yes, you have missed the previous codes because while you might have not run the run UMAP command. That's why you're not getting UMAP in the Sirat cluster. So you will have to run this command. I'm pasting right now. I'm pasting right now. Yes, run this command. Okay. So guys, this is all about this our single cell analysis as of now, and there's a lot of downstream analysis based on, but this is the basic, and good start for the single cell analysis. You start with the cell types, then cluster them, then identify the marker genes, their differential expression, then assign the cell types, then cluster them. Okay, so now floor is open for the question. We can have the open discussion. You guys can ask me your doubts, I can answer. I hope you have run this script, run this analysis. I can also tell you one very important thing which you should always follow while doing any such analysis, that you should always do that. Okay, so let me So guys, whenever you're doing any such analysis, try to save your file as an RDS file. Okay, so here I'm saving this as RDS file. So if I will save this file here, so next time I don't have to repeat anything. I can directly, suppose for example, I have saved this object as this RDS file. So I will show you that how this is going to save my time and my analysis. Okay, there's one. Which is with the PBMs. Okay, Salmia, the PBMC one was just just testing. How do we do the analysis for a specific gene instead of the whole set? How can we do the specific gene? You can select the gene. Okay, so what? Why do you want to do the selected set of genes? I don't know. Aishwarya, Murli, could you please explain your question a bit better so I can understand? Okay, so since Aishwarya will take time to explain the questions.
Can you
Yes,
hear me?
Aishwarya. Yes.
So, if I want to study a specific set of genes and their differential expression across a particular single cell RNA-seq data set, so where do I choose the genes which I want to study instead of the whole set?
Okay, so you can do one thing. You can select the genes like the expression plot I was showing to you. Okay, so let me show you the expression plot. My God, this is not going. Okay. Why when we can expect this code? Yes, I will send to your organizer right now. This is not too much. Okay, so sorry for your answer is that you can select the gene on the basis of genes by selecting the cell types where these genes are expressed. Okay, and second thing that you can check the expression dynamics of your selected genes in different cell types. So suppose, for example, you are interested in you have the list of genes which are responsible for the metasis. Okay, and so you can plot the expression of your selected genes in the all the cell types present in your data. First thing. Second thing, you can also check the expression of those selected genes. Okay, when we were doing the find all markers, so this function would allow you to get the expression of all these differentially expressed genes. So you can check whether your select set of genes are differentially expressed in which cell types and which cell types they are highly expressed. Okay. Second thing that if you are comparing one versus other condition, then also you can check the expression of your genes in particular state by comparing the two conditions.
Yeah, but my doubt is like at what point in this R code I can choose the genes, like when the Seret object is being generated.
So basically, see, once Seret object is done, so you can do the visualization and analysis any point of time after this annotation.
Okay. Yeah.
I want to ask what basis you cluster mean the cluster 1 can be any cell types, but why you named it? Okay. Okay, Divakar, I named cluster 1 as a cluster 1 as a naive CD4 on the basis of expression of the marker genes. Okay, so really, if you ask me to name the markers, exact markers right now, I don't remember, but it is based on the expression of marker genes. For CD4 markers, I have to check what are the CD4 naive CD4 markers, but yeah, that is very because that was some literature based. So yeah, definitely it is based on only on the expression of marker genes. Naive CD4 marker genes are present in the cluster 1. That's why. Highly expressed, not even present only. So guys, see what the main important thing I was about to tell you people that since once all the analysis has been done, so you can save these analysis, this object as a one RDS file. And next time, whenever you want to proceed with that, so you can just read this file. Okay, 1 weekend. So there is a set of lot of repositories are there where you can get markers for different genes. Okay. Second thing is that there is something called automatic annotation, like automated annotation. So single R is one package which you can try to annotate your data cell type. And most of the cases, it annotates cell types present in the cancer samples. It might be a bit problematic when you're analyzing some neuro data. So single R are such tool where you can annotate your cell automatically rather than going and checking this differential expression of each gene type. But yes, even annotating in different these default softwares like automated software, you have to go back and check the expression of your marker genes. We can do it automatically also with like single or some other tools are also available. But again, we have to confirm the expression of marker genes. So yes, so see, I saved it, and now I read it, my whole object in a new file. And if I will want to do any sort of present, suppose you just save this file anywhere, and you come back from holiday, you come back from after 6 months or 1 year, then you have to just load this file and you can do all the analysis like here. Okay, so just let me show you. Yeah, okay, you get the same plot. Okay, so don't forget to save all the your last object in the as a RDS file, because this will save your time. Okay, so now I stored it, and I stored my, again call out my object, and you can see I don't need to repeat all these steps again. I know what are the last markers and what are the last labels I assigned to my clusters. And now you can see I can easily plot myself. I can easily plot anything. Even I can show you, we can plot, in fact, everything. Oh, let me show you some important thing. I can do that. For example, this one. So like here, I want to check the expression of these genes in my data. So if you see, these MS4, CD9, 17 are highly expressed in case of B cell. Okay. Similar, any markers you can check. I can check these markers, suppose these set of genes in my object. So yes. Okay, so if you see the expression of these genes, this is B cell. Okay, so if you see that MS4 is highly expressed in B cell, then yes, this is the gene which is highly expressed in B cell. For GNLY, this is expressed here. And what are the cell types? You see, this is NK. Okay, so guys, I hope okay, so it's come now. I think from my side is completed. Now floor is open. You can ask any question. You can stop. I can stop presenting. I will share this script with you people. That's not the issue. Okay. So is there any question? Anyone? Okay. This is the RDS format or the structure of these files. And these files will contain, this object will contain your different parameters and your, okay, not RDS. Please, can you show your screen a bit?
Asking about, and Salim, I was just asking about the 3 files initially we started with, and how is the data structured in that?
Okay. Yes. Okay, now it's screen visible. Someone was asking me to put it. Okay, Salim. So Salim, initial files are mostly in the matrix format. Okay.
Yeah.
In an initial files are matrix. Okay. And then barcode files, and then this D file are TSB format file. Okay, so matrix is like a count matrix. Okay, so you will have a feature versus cell matrix. So where in first column you will have the features, and then in the column you will have the cell. Okay, so each cell type will be have the unique barcode. Okay. So, and in addition to that, you will have the barcode information in the barcode file .tsv format, and also the features information in the gene TSV format file. So one is count matrix, one is the information for your features, one is information for your barcodes. Okay, initially these are the files we use. Okay.
So for each feature, how frequently it is expressed in a cell, that will be the count.
Yes.
I'm actually new to this field, that's why I'm asking.
No issues. Feel free to ask, guys. Feel free to ask. This is all my purpose is to make you people comfortable in the analysis. So don't hesitate. It's nothing like new or older in the field. No, nothing. Yes, please feel free to ask. Yes, I would be happy to answer. Yes, can you please? Okay, I have done that. 1 minute to finish scripts. Okay, how to get the markers names from the, can you chase the sites? Yes, there are a lot of sites where you can get the markers. I can say those sites, don't worry, where you can really get the markers for each cell types almost. One tool you can refer, single R, is that, which helps you for the automatic annotation. And another thing is that even in Serrat, there's a level transfer something. Can you suggest some art or more art? Yes, Diti, I will share some art tutorial where you guys can get. How did you divide the data matrix into separate object on the basis of specific? How did you divide the data matrix into separate objects? We are not dividing the data matrix into separate object, Prakriti. I couldn't understand your question, I think. Prakriti, could you please explain your question a bit more? Okay, so guys, shall we in the workshop? Do you have any other question? And I would, hello, can you just explain how flow we just perform? Yes, I will say. Okay, I will do that. How to divide the object using that. Okay, subset. I think, Prakriti, are you asking about the subset? What are all of the packages? Okay. For differential gene expression analysis, there is a package called DSEC, Lima. You can use AZAR the package, but DSEC, I think, for in case of differential gene expression from the transcriptome data, DSEC is a good one. Which resource do you recommend for a beginner to learn how to write scripts? Okay, yes. So you can refer such tutorials, such workshops which we are doing right now. So the good thing is that to motivate yourself for this such any analysis, once you do the analysis, you get the output that fascinate you. Okay, so like at the end of the workshop, if you are following this and you were getting this such nicely labeled plot, like B cell is there, NK is there, DC is there, palette is there. So these are the things motivates you, and I would recommend you to attend such workshop more frequently and try to follow what we have done here in your own computer, in your own space, and by yourself. So that will help you. Okay, Prakriti, yes, subsetting the object, you can subset the object on the basis of criteria you want to subset. For example, if I want to subset for the palette, so I will select the column of this my idents, and then I will give the name of this palette and subset out the specific cell types. So we can subset that, but you have to provide the column name and the ident you want. Like the column name and the cell types you want to subset. You can do that. Which source can you tell me the sites? Yes, there's a lot of sites I can tell you. This single R is one of the most comprehensive, package which is for the automatic annotation and having all marker genes also. You can go. And for Monjusa and the people, those who are asking for the marker genes, I would suggest you people to go and read any paper, paper published in high impact factor journal, where people have done the single cell RNA sequencing, and for the different samples and different tissues. There you will see the marker gene list in the supplementary files. Okay, so that is another way to get the marker genes, because people provide marker genes for each cell types whenever they annotate any cell types. So the second thing, one thing is that you can go to single R or some other packages. Another thing is that you can go to the paper, and in the supplementary section, people provide the marker gene list. Yes, can you please explain about the? Okay, Venkatesh. Yes.
Yeah. So when you use the marker genes for Insurat, do you always supply your set of marker genes, or use the one which is there
No. So see one thing. Okay, so in Serat, you don't need to supply your marker genes. Okay, what you do exactly, you check the expression of your marker genes. Okay.
Okay.
Okay, suppose for example you have marker gene AQP4 for astrocytes. Okay, you know that.
Yes.
And you have the scRNA from the brain samples. So, and these are clustering into different clusters. So first job is that you will check the AQP4 expression in the cell, in the markers, okay, in the different clusters. If you see for cluster 1, the AQP4 expression is very high in the cluster 1. This means that the cluster 1 is the bunch of my astrocyte cells.
Okay, and I need to have the my previous information in hand, then check the expression, and based on that
yes
cluster
then you can assign the identity to that cluster.
Identity of the cluster.
Yes.
And also, how did you derive these 3 files? From the analysis pipeline you showed initially, this matrix and barcode, all the 3 files?
Yeah, so this is output of the Cell Ranger file, Cell Ranger, the 10x output. Cell Ranger, this is software which.
Yeah, in 10x Genomics, that's Cell Ranger software. Okay, you use the raw file, and this is the output. These 3 files are the output from Cell Ranger.
Yes.
Okay.
Anyone else have any question? Please let me know, please, guys.
And you have also mentioned about just before we started, you mentioned about deconvolution.
Yes.
So how good does it reflect if we deconvolute and calculate the cell type proportions? How close is it to the clinical analysis? What's your take on that?
So what is doing the deconvolution? That you have set of genes. Okay. So whenever you are doing deconvolution, you have to supply the gene versus cell
what I'm asking is how close do you think? What's your take? I've done deconvolution, and then I was there's not enough data to exactly compare and see if I do single cell and if I deconvolute my bulk data, is it how close they are comparable? That's what.
Yes, it is very pretty close most of the time. If your tissue is coming from the same samples, so these are pretty good techniques, and it gives you a lot of confirmation also.
Okay.
Anyone else? Selby, is there any limitation known for SIRAT package and single cell analysis? Yes, there's a lot of limitations are known for the single cell analysis. One of the is that you won't get the expression of transcription factors. This is the limitation for single cell analysis. Then limitation for SIRAT package, yes, sometimes SIRAT doesn't do the proper clustering. It fails with the larger number of data. Second thing that, sometimes you might not able to find the differentially expressed genes, which is actually differentiating the 2 clusters. So these are the challenges we are facing in SIRAT. But yes, people are working on that. But still, I would suggest SIRAT is better than, the competitors. good one for me at least. Anyone else? Thank you guys for it. It's been almost 3.5 hours. Okay. So shall we close? Do you have anyone have any questions? So please let me know. Otherwise, I will conclude the session. Okay. Okay. Thank you. Thank you. Thank you guys for your feedback. I would love to hear. Yes, I will share the resources. I will share the resources. Any study groups for beginner? Yes, yes, this data, PBMC data, which we are using right now. Silent can be used as a beginner. Yes. This data. Yes, thank you, Lukni. Thank you, Shweta, for this wonderful feedback. Thank you. Thank you. And thank you, Sangha. Thank you, Helen. Thank you, Julie. Please forgive me if I pronounce anyone's names wrong. Thank you, Moyo. Yes. Okay, can you? Yes, yes, Amit, I will say thanks, Venkatesh, for your feedback. Thank you, Amit. Thank you. Thank you. Thank you, G. Thank you, G. Oh my God. Thank you, Vinodni. Thank you. Okay, guys, I think, yes, thank you, Anusha. Yes, we can. Yes, we can. I will share it with your organizer. Okay, and then you can get it from them. Thank you. Oh, yes, yes, let's get. Thank you. No issues. Always pleasure to share knowledge and share expertise with people. Yes, thank you, Vignesh. Thank you for the feedback. Okay, guys, I think we can close. Okay, so thank you so much, everyone, for attending and for your patience. And I'm happy to see that not many of you, but yes, at least some of you tried and analyze the data. That is very important for me as a tutor. Whenever your people are trying in their efforts to analyze the data, that is really pleasurable for me. So yes, what kind of tissue we can choose? I think as of now, people have tried single cell of all the tissues present in our body, I think. So there is no limitation of the choosing the tissue for the single cell analysis. You can do single cell analysis for any sort of tissues as per my knowledge. Okay. So thank you, guys. I will share this recording and script to your organizer. Okay. Thanks for attending this workshop. And it was I really enjoyed a lot. Okay. Bye-bye, guys. Good luck.